Hello, everybody. Thank you so much for spending time today with us and allowing us to explain how to achieve high-quality data in highly multi-parametric flow assays, data that can be used in various research fields, including the evaluation of the immune response to various diseases like COVID-19. In today's webinar, we're going to briefly talk about the technology that is used in our instrument, and then we're going to spend most of the time talking about the application side and panel design. This is the key to get successful data and data that is high quality. And we're going to use a 36-color panel that I developed as an example on how to apply the best principles and the best guidelines to get to high quality data. At the end, I'm going to briefly show you tools and collaborations that we have going to get the most out of the data once we know it's robust in terms of analysis. And I would like also to tell you a little bit on how we would like to work with you and how we would like to make things happen in your lab. Let's start by briefly, maybe some of you are new to the Aurora and what is different about the Aurora compared to a conventional cytometer. So let's start with a quick overview of what is the full spectrum cytometry approach. So every flow cytometrist in the world has used at some point or every day PE in their flow cytometry assays. In a conventional cytometer, what you have is a detector that is going to be the PE detector that will have a bandpass filter that has been designed to cover the most of, or in the entirety, the peak emission coming from PE after PE is excited by either the blue or the yellow-green laser, depending on the instrument that you have. As you can see here, you're going to get a lot of the photons coming from the PE excitation, but there are going to be a lot of information also that is not going to be captured by any detector, that is going to be ignored. And there is going to be some light going to detectors that are assigned to other dyes because of the emission is broad, that is going to be mathematically corrected using compensation that I think many of you in flow cytometry are familiar with. So now let's look at what happened with all the light coming from the PE excitation in a spectrum cytometer. In a spectrum cytometer, and the Aurora is a spectrum cytometer, what we do is something different, and is that we are going to take many slices, and we're going to cover the whole emission spectrum of PE. We're going to try to get as many photons as possible that are coming after the PE excitation. This is an example of what happened of the emission spectrum when PE is excited by the blue laser. And what you're going to see is that through these multiple slices, we're covering the full range of emission, starting a little bit around 550 nanometers and going all the way down to 700 plus nanometers. You're going to see that there are two areas where we have a gap, and that gap is due to the fact that we have to skip the laser lines that are in the instrument. In this case, in this instrument, if we have a yellow-green laser or a red laser, you will have to skip those. And what is very interesting is that while this is done for the light that is emitted after excitation by one of the lasers, in this case, the blue laser, we are doing exactly the same thing if the dye is excitable by other lasers in the instrument. So in the case of PE, if you have an instrument that has a blue, a yellow, green, and a violet laser, in those three lasers are going to efficiently excite PE. And the emission coming from that excitation is going to be detected through the entire range of emission by detectors that are seeing the light coming from each of those three lasers. And this is why in the Aurora we talk about an approach of full spectrum cytometry. So in order to make sure this is clear, let me show you a little bit more in detail what happens in the instrument, what is the optical design. We have made every effort and we have been using state-of-the-art technology to make sure that we see every single photon that is emitted after the excitation of the various fluorochromes, because that will ensure that you have the best possible data. Here is the optical bench of a 5 laser instrument, and you see we have 5 lasers, and each laser is specially separated. Each laser has its independent optical path, and each laser is linked to a module. In those modules, in those detection modules, is where you have those multiple filters that I explained in the previous slide, and that are going to allow us to detect. Those filters coupled to high sensitive detectors will allow us to detect the full range of emission in every laser, starting a little bit after the laser line and going all the way to above 800 nanometers. Why do we stop at 800 nanometers? Because we don't have currently, we stop around 810 nanometers. We don't have dyes emitting beyond that. If there are more and more dyes that will be available going further in longer wavelengths, we have the possibility to also capture that emission. So it's a system where every part of the optics has been carefully thought and worked on. And now I want to show you what happened when a particle goes through the system. So remember, we have these very special detector modules. And here is an example of a particle that went through the system. That particle was labeled with a specific fluorochrome. And what is going to happen is that through the excitation with multiple by each of the lasers, we're going to detect the signal, the MFI, that is the intensity represented in the y-axis of this plots that just appeared. The emission or intensity through the different detectors that are in the x-axis. So you're going to see that there are in some detectors we have very little light, very little intensity. In some we have more light. And these ups and downs are going to tell us which detectors are seeing more light depending on the wavelength of emission, and which ones are not seeing any, because at that wavelength there is not really photons coming from the excitation of this particle labeled with this particular dye. Now, lots of people ask us, what are these colors, this red, green, and blue? Think about it like if you were seeing a histogram from the top, and the coloring is like a density plot where the red shows you where we have the majority of the events, and then it goes into a gradient color until blue, where blue is where we have the fewer number of events for that distribution in that specific channel. The software then is going to do something very special and is going to stitch the information coming from each of the detector modules. And what we're going to end up is, as I explained, with something called the full spectrum signature. What the full spectrum signature is in the x-axis we have every single detector that is in the instrument. In a 5 laser system, we'll have up to 64 detectors. And in the y-axis, we have intensity, MFI, light. And so the full spectrum signature is all the information coming from all the detectors across all lasers when a particle goes through the system, because we're going to see what happened when every single laser excited that particle. The full spectrum signature is the foundation of everything that we do in our system, and understanding how they are generated and understanding what it means is going to guarantee that you have a successful experience in our system. Now, what happens if we run a sample that has multiple dyes on it? So in this case, I'm showing you a full spectrum plot that has a mixture of signatures, of full spectrum signatures. And so the combination of the optics are allowing us to see all this information. And now we have to apply some math to it to be able to tell if a particle has multiple dyes attached to it, what is the contribution of each dye? And we do that by a mathematical process, and that is used not only in flow cytometry, but in other domains. that is ordinary least squares that calculates the contribution of each known fluorochrome to what we are seeing here as the total spectrum that is generated by a mixture of those. So again, in this spectrum plot, what we have is a mixture of spectrums. And then the question is, how are we going to know what is the contribution of each dye? And then what happened is that we're going to use a mathematical procedure called an algorithm called an unmixing algorithm that is a linear regression called ordinary least square that is going to calculate the contribution of each signature of an individual fluorochrome to this mixture. So think about it as if you are extracting from this mixture each component, and for that component, the software needs to know what is the signature of each component individually. So at the bottom, you have a plot that has a mixture of colors, and if the software knows what is black, it will take out the black, the red, the green, the purple, until nothing is left. It's more complicated than that, and there are a lot of good information out there on what exactly linear mixing is, but this goes beyond the scope of this webinar. But if you're interested, please make sure to reach out to us so that we can explain to you more and direct you to the resources to fully explain what is unmixing about. So, what is a typical workflow on our instrument? So first, and we're going to spend time talking about this when we talk about assay optimization, we need to acquire controls as you do in a conventional cytometer. We need an on-stain control, and we're going to explain why this on-stain control is important to us. And we're going to acquire controls that are labeled with each one of the fluorochromes that are present in our sample. Those are called reference controls. The user interface and what you're going to see while acquiring those controls is very simple. And basically what you're seeing is the beautiful spectrum plot and histograms showing you what is going on through every single detector that is in the instrument. Then you're going to click on mixing. As you click in our softwares on calculate compensation, here you're going to click on mixing, and beautiful wizard is going to be opening in front of you. We have put a lot of effort in this wizard to make it as robust and as informative as possible. We're going to assess, and we're going to make sure that each control that you run has the right information for the unmixing, for the mathematical deconvolution of the data to be accurate. And once that is done, that is a very simple process, then you're going to run your multicolor samples. The unmixing is going to happen real time. And what you're going to see display in front of you now is, as you normally see your beautiful multicolor data, you can draw plots, you can draw gates, you can play with your data while the acquisition is happening. If you have, if you want to get more details and if you want to see how to run samples, please check our YouTube channel. There is tons of useful tutorials and videos in there. Okay, so I've explained to you that we have a different technology, we have a different way of treating light, we have a very special optics design, and that we use a mathematical approach to use all the information that is coming from the instrument. So why is it important, and how are we using this technology to enhance our research and the data that we can obtain from our flow assays? So the main driver for us, and what is most important to us, is that you get high quality data. And so here I wanted to show you quickly an example when the same assay, exactly the same assay, is run on our instrument and on a conventional cytometer. This is a table that you're going to be very familiar with if you start working with us. We like to present our panel designs in this table, with every row correspond to an approximate emission wavelength, and the columns are each laser and the dyes that are primarily excited by each laser. And then what you're going to see in this panel design, if you look at the violet column, is that we have many dyes that are excited by the violet that are close in emission. Now, in the conventional cytometer, depending on the instrument that you have, you might have filters for the detection of each of these dyes. So we designed an experiment that was theoretically being able to be run either in the conventional cytometer or in our system. And so I want to show you the data. First, and remember, we run exactly the same assay, meaning that we run exactly the same single stain controls in both instruments, and we use FlowJo to calculate the spillover spreading matrix. At the top is the matrix coming from the data generated in the Aurora. At the bottom is the matrix coming from the conventional cytometer. And without going into the detail of the numbers, just by color coding the numbers, high numbers are in red and mean there is spread happening. White or light pink means there's not so much spread going on. You can easily see that there is more red and bright pink in the matrix at the bottom. That is the one from the conventional cytometer. Now, what does that mean, and do we really care about the spread and these numbers being high? How does it translate in real data? So here is the data, and we plotted the data on FCS Express, and we made sure that the data at the top and the data at the bottom was gated identically and that the same scaling was applied to both sets of data. And what you can see is that if you take a given plot, any given plot, you're going to see that the negative populations, and this is something that I'm very strong about, and some of you, if you have already heard me talking or if we have been in your lab, we're very, very enthusiastic about using the negative populations as a guideline of how good is the data. When the negative population is nicely clustered, you're guaranteed to have good data in front of you. And what you're going to see as you compare all these plots is that with the Aurora and using the spectral approach and the unmixing, we have very high chances to get nicely clustered tight data. Whereas with the conventional cytometer, because of the approach, because of the optics, and because of the use of compensation, you're going to get more spread, and your ability to see dim populations is going to be somehow compromised if you're using dyes that are highly overlapping. So the ability to effectively use dyes to get high quality data, even with dyes that are close in emission, is a huge advantage and something that we strongly believe, and we believe is the way to go in flow cytometry. Okay, so remember I told you quickly in the workflow that we needed to run an on-stain. And some of you in conventional cytometers, you like to use on-stain because it's part of your setup strategy. In a spectrum cytometer, the negatives are key. Why? Because when you run negative cells, those negative cells are going to have their own spectrum. As we have the spectrums of fluorochromes, we have spectrums of on-stain particles, and that spectrum corresponds to the autofluorescence of that particle. Here are various examples coming from different sites that we work closely in collaboration with, showing you that the level of autofluorescence and sometimes even the autofluorescence signature is different depending on the particles or sample types that you run in your lab. And we can use this information as part of the information to assess what is the information in your multicolor samples. What does that mean? Here is an example of data from a collaborator in Germany, and he was very interested at detecting M. Sherry in a specific biology system that he was using in his lab. As you can see, the on-stain in his cells were already having high signal, high intensity, meaning high autofluorescence. And there were areas where the M. Sherry signature and the on-stain signatures were somehow very close. So what happened when we run the assay? We look at the on-stain cells and we look them at the M. Sherry dimension, and we see that there's a lot of signal going on already. Why? Because the cell had high autofluorescence at that wavelength. Now, If we run the cells that were labeled with the M-Sherry, you're going to see that, yes, there is a signal, but this is one of those situations where I always telling people that if you put multiple people, flow cytometry experts, to gate on this data or to judge what is going on here, lots of people are going to have different ideas. Some people will tell you this is noise. Some people will gate in a very different manner to see what is positive and what is not. What happens if we have the ability to take away the autofluorescence and separate the signal coming from the actual emission of the M-Sherry? Magic. Not really magic, but it's really mathematics and photons. Now, with this system, we have the ability to say what is the true M-Sherry signal and to take away the autofluorescence of the cells. And now we can focus again on the real, what is real, and the biology of the assay. How is this possible? Using spectral analyzer and using a highly sensitive spectral analyzer like the Aurora. Okay. As I said as well, the full spectrum signatures are the foundation of everything we do. We are very excited right now to work with a dye that many of you are not familiar with. It's called CF568, and we have launched our first reagent, a CD4 for human clone SK3 labeled with this dye. If you go to a vendor reagent vendor website and look at the spectrum of CF568 and P, you're going to see no way. Those two dyes are very close together in terms of their emission spectrum. However, as I mentioned to you, let's not focus on what happened through the excitation of one single laser. Let's not focus on what happened on the emission in a very specific area of emission. Let's look at the full picture. Excitation by multiple lasers, emission through the full range of wavelengths. And what you are going to see is that although PE and CF568 share some similarities in their spectrum, they are also differences. And we can take full advantage of those differences because differences mean that these two dyes are actually distinguishable because of the optics and because of the unmixing algorithm. And so here is an example that of an assay that we have been using where we have CD4 in CF568, and you're going to see that the CD4 versus CD8 plot is beautiful. We have very nice resolution, and out of the CD4s, we're looking for Tregs. We're looking at CD127 versus CD25 PE. And guess what? We have a very nice resolution of the PE. We are not bothered by the fact that these two markers are co-expressed and were assigned to similar dyes, but differently enough because of their full spectrum signature. Very, very exciting. Okay, to visually compare the signatures was something that we were doing early on when we launched the Aurora, but many of you were asking, how close to us can two spectrums be before I call that those two dyes are the same or identical? And so we hear you, and our mathematician and software team has worked at developing a metric, and we call that metric the Similarity Index. What this is a way to compare the full spectrum signature of two dyes. The index ranges from 1 to 0. 1 means that we have exactly the same spectrum. 0 means that the two spectrum are completely different. If you are interested in the math and how we calculate this similarity index, please do not hesitate to reach out to us. I don't have time today to explain to you, but it's we have lots of data, and we are really, this similarity index have opened the door for us to explore more and more dyes, because now we have a very robust metric to assess what is the same and what is not. So let's look at two dyes. PerCP E-Fluor710 is a beautiful dye from Thermo Fisher that we really like, and then PerCP Cy5.5 is one of our more classic dyes that many of you use. If we look at their spectrums, you are going to see that they don't fully overlap. There are similarities, but again, there are areas where they are different. What is the similarity index for these two? 0.9. And we effectively use these two dyes in combinations in our multicolor panel. Now let's look at another example, a dye excited by the UV, BUV 395 and APC that everybody is familiar with. You're going to see that these two curves, the gray and the red, have 0 areas of overlap. They're absolutely different. And guess what? Their similarity index is 0. So here we go. We have a nice metric. We have a number that we can provide you, that we can tell you these two dyes can be used in combination, yes or no, depending on their similarity index. So now that you have a big overview and understanding on how we use the technology and why we are so excited about it and why we can do things differently than what has been done until now in flow cytometry, let's go to what is very important in your lab when you have access to this technology, and is to do a very thorough panel design. We receive a lot of requests about panel design, and my answer to many of you and colleagues who come asking for help to design a panel is, this is a biological assay, and the foundation and the only way to get data is to know as much as possible about the biology. This is where everything starts. So we need your help to gather as much information as possible around the biology of the assay. Or when I am designing a panel, I need to spend time reading, researching the markers, understanding as much as I can of the biology for every single marker that I'm going to be using in my assay. What are the two key pieces of information that I need? I need to understand what is the level of expression of each antigen in the cells that I'm interested at analyzing. So I'm not interested in the number of receptors per cell. I'm interested in a broad classification, and we're going to talk very briefly on how we do that classification. And the other piece of information that we need is antigen co-expression. Now, lots of you might be thinking, there's no way I can find this information. This is too complicated. It is not. You will be surprised on information that is already available in your lab, in the literature, through flow cytometry reagent vendors. There's tons of information out there that can easily help us to get these two pieces of information. When it comes to classifying the antigens, basically what we're going to focus on is 3 groups. And think about, if you are drawing your gating strategy, what are you going to use at the very top to identify the main populations? Those are the antigens that we call primary. Which ones are coming next in your gating strategy? Those are the secondary. And then which ones are at the bottom, which we call the tertiary. This is not something that we came up with. This is something that has been was published by the Roederer lab, and that I think is heavily used and accepted by flow cytometrists in the world. In the assay that I'm going to walk you as an example is this 36 color panel that we are currently using in many different sites. And what I did was to identify my primary antigens, and those you can clearly see have usually a bimodal distribution, black on white, and these are examples of the primary markers in my panel. I have secondary markers that are going to allow me to further subset, for example, the T cells, the B cells, the NK cells in different stages of differentiation. And you are going to see that those markers are very often expressed as a continuum, as they are expressed in different levels depending on the level of maturation of the cells. And then I have those markers that are critical for me because either they're very dim or they're expressing very rare populations, and I need to make sure that I obtain the best signal out of them. Those are called tertiary, and here is an example. For example, PD-1, that depending on the donor, I know that in certain diseases the expression will be super high, but in normal donors we don't have such a high expression, so I classify it as tertiary. So with this already in mind, plus researching the literature and understanding what markers are going to be key at subsetting the cells of interest, I came up with this cartoon where I know exactly what to expect in terms of co-expression for different subsets of cells. And so if I show you, for example, what I'm going to be doing with the CD4 T cells, I have a very clear understanding of what markers I'm going to be looking at, and that all these markers can be co-expressed on the same cell types or in different subsets. And I need to use that information when it comes time to panel design. So again and again, the biology is the foundation, is the starting point. We have knowledge of certain areas of biology. If you have come with something that we don't know, we are going to encourage you to spend time together researching and finding this information before we do anything regarding panel design. Okay, what can we help you with, and what do we have a lot of information and we can provide to you as a starting point in terms of the panel design is the fluorochrome selection. To choose which fluorochromes you are going to include in your assay, we are going to take care or think about three main aspects. What is very important, as I already emphasized in the example of the CD4 versus the CF568, is that we care tremendously about the full spectrum signatures. The full spectrum signature is everything, it's the fingerprint of the dye, and we really need this information, and we use it in many different ways. As I show you, one of them is the Similarity Index. We're going to see other aspects of it that we use. As in a conventional cytometer, we care about the brightness of each fluorochrome, and we care about the spread, how much spread those dyes can introduce in other dyes. And it's the combination of these three pieces of information that is going to allow us to make the best possible decisions when it comes to panel design. Okay, so we have out there, and we have put a tremendous amount of work with our R&D team, our marketing team, our software team, a lot of resources when it comes to fluorochrome selection. We have what we call a beautiful document that is available through our website or in our software called the Fluorochrome Selection Guidelines, where you have the signature of every single dye that we have tested. Online, we have a spectrum viewer that is very, very cool. And we also have online, you have the ability to create a similarity index, and is also a tool that we use in our software when you're going to unmix your experiment. So tons of tools that we have developed to make sure that you fully understand the signatures, and that one day, hopefully, you memorize as many of these signatures as possible. Concerning the brightness, we test every possible reagent dye that we can have access to. We try to get these different dyes in human anti-CD4. We try to go for the same clone. It's not always possible. And we run it on the same instrument, and we classify the brightness based on stain index, which is the metric that is in the y-axis, is stain index of the CD4 stain cells labeled with these various fluorochromes. And what you're going to see is that we get a full range of brightness from dyes that are dim, dyes that are intermediate, bright, or very bright. In the panel that I am going to design, I am going to be using a variety of dyes that span the whole range of brightness. Why is this important? Using exclusively dyes that are very bright is going to be challenging, as those dyes, I always use the analogy, are like this very loud neighbor that is really difficult to ignore. It's going to be difficult to ignore what? The spread that they're going to be introducing. But we need them. We need them, but because we need a lot of good dyes, bright dyes, to resolve dim antigens. At the other end of the spectrum, we have those dim dyes that I really, really like, because if you have a system with high resolution like ours, those dye, even if dim, are going to give you very good separation. And they're quiet. They are not going to disturb that much any other dyes, and they're not going to introduce a lot of spread. So we really like, and I think a good panel design is a design that balances and uses dyes across the whole range of brightness. This table shows you the 35 dyes. There are 35 fluorochromes and 1 viability dye, the live dead blue from Thermo Fisher, that we really strongly recommend using if you have a UV laser. It's a fantastic dye. And you're going to see that we are using a variety of dyes and a range of brightness. In this table, the number of dots correspond to the level of brightness from dim to medium and high brightness. And so we have spent a tremendous amount of time understanding which dyes will work well in combination, not only because of their signatures, but also because of their brightness. And now comes the third aspect. So first was the fluorochrome signature, then was the brightness, and now we're going to talk about spread. This is the spectrum signature of two dyes, PSI5 and APC, and you're going to see that there are areas where they have high overlap. In this area in the red laser, and some people do not like to use PSI5 because it almost behaves like APC. If I overlay these two curves, you're going to see that there is a lot of the area underneath the orange curve that is occupied by the blue curve, that is the PSI5 dye. So what does that mean? It means that when we unmix, when you compensate, you're going to have a lot of spread of the PSI5 into the APC direction. Okay, do I know, is this a surprise? No. How could I anticipate that? By looking at the signatures. It's very clear that the two dyes have overlapping emission in the same area. Are there ways to measure how much the spread is going to influence the resolution? There is. There are multiple methods. You have the SSM that I already talked very briefly when comparing in that initial comparison that we did. We have our own metric that is called the cross-stain index matrix, that is just looking at changes in stain index from when the dye is by itself. Like this is the cross-stain index of the fluorochrome Y when it's by itself. And now if we introduce fluorochrome X and fluorochrome X introduce spread into Y, that stain index is going to be reduced. So we use this as a metric to guide us and to make us understand, help us understand what is going to be the impact of spread. If you want to have details about what the spread matrix is, how it's calculated, please visit our resources page in our website. Under resources white paper, we have a new white paper that is really outstanding, and it will give you tons of information. At the end of the day, what we generate are these heat maps. Again, as we were discussing with the SSM, red or bright pink means there's a lot of spread. White or light pink means there's little spread. What does that mean in terms of data? So in this example, APC and BUV 805 have little spread between them. And see, if I use those two dyes, if I assign them to markers that are co-expressed and where the expression is as a continuum, I get optimal resolution. I can see everything that is bright, that is dim, intermediate, bright in both axes without any problem. Now, let's look at one of those combinations where we're a little bit concerned because it shows in red. This is FITC versus a Spark Blue 550. Spark Blue 550 is a new dye from BioLegend that we really, really love. It's close to the emission of FITC, and yes, there is spread. Is that the end of the world? No. If we know and we have paid attention and we know there is a spread, in this example, we use those two dyes assigned to antigens that are not co-expressed. And so we are not compromising our assay. What is another tool that you have? Because I know some of you will tell me, Maria, I don't have the luxury of having all my antigens expressed in different cell types. Everything is co-expressed. Well, you have another thing that you can play with, and it is the level of expression of your antigen. Because if you use something in FITC and something in Spark Blue that is expressed at low levels, then you are going to have little spread, and that is going to facilitate using these dyes that are highly overlapping. So in terms of metrics, because everybody would like to have a definite metrics, is this dye combination going to work or not? We have developed another metric called the complexity index metrics that you're going to find at the bottom of the table where we have all the similarity indexes. Where can you get this? Through our website, again, or in the software when you're going to unmix the data. And basically, the lower this number means the higher likelihood that you're going to use dyes that are not going to introduce a lot of spread into each other. Here are 10 dyes that work really well together. We have a complexity index of 2. And in the 35, 36 color panel, we can have that index going up significantly. Why? Because we're using dyes that highly overlap. Regarding the complexity index is also a great tool and something that for education purpose, I would like you to start thinking about when it comes on the shape of the negative cells when you use high overlapping dyes. FITC versus FITC is the same dye. If you try to unmix this, we are going to get a diagonal. It's exactly the same dye. FITC versus Alexa 488, FITC and Alexa 488 have a very high similarity index. They are almost the same. There are very little areas of difference, but it's not enough to resolve them, and we do not recommend using these two dyes in combination ever. See, you have a very high complexity index if you think about only two dyes. Now, FITC and Brian Blue 515, we have heard some people questioning, should I be using those two dyes together? They have a similarity index of 0.99 and a complexity index that is 9. And for us, this 0.98 similarity index and this complexity index of 9 is good enough, and we get good results when we use them in combination and we follow panel design rules. We can resolve them. The negatives are the slant, which is expected with the level of overlap of those dyes. But with good panel design, assigning it to the right markers, you can use these two dyes in combinations. FITC and Alexa 532, no problem. Our complexity index goes even down. FITC and APC, a walk in the park. So the complexity index tells you a lot, and is going also to tell you, because of the overlap in those dyes, how are your negatives going to be behaving. When you look at the complexity index in the matrix that we're providing, only look at half of the matrix because the other half is exactly the same information in duplicate. So we will be working at correcting this in future releases. And here is using the information in terms of spectrum signatures, brightness, and spread. The panel design that I came up with. As I said, presenting the panel, when you ask me, please, Maria, could you review my panel? I will do it in a heartbeat. Please help me. Laying it out this way, as I explained, each column being the dyes primarily excited by each laser, the rows being the emission wavelength. This will allow me to quickly QC the data. I will look through columns, what happened with the dyes that are very close in emission, what antigens were they assigned to, and I will also quickly look at the rows, because in the rows you have the same emission wavelength, and if those dyes has cross laser excitation, which is almost always the case, I want to know what is going to happen with the antigens that are in that line. How can I gauge this? If I have the biology information, how are CD25, IgM, CCR5 going to behave across the cell types that I'm examining so I can make the best possible decisions? So putting all the information together from the floors and the biology and displaying the data like this, you are going to quickly have a good assessment if your theoretically panel design is good or no. Now we need to test it. And unfortunately, I'm running out of time, but in order to get good data, the only possible way to achieve this in a conventional cytometer or in a spectral cytometer is to have good controls. How good? The best possible controls that you can. Don't take shortcuts. We need to make sure that we follow the basic rules that every flow cytometrist know, that the controls are well separated, that we collect enough events, and that the control is a good control for the spectrum of the multicolor sample. In the latest version of the software, we use the similarity index in a different way to the way I have explained until now. You can save in the software what we call a benchmark, And we're going to compare every that if you save the FITC benchmark, we're going to compare every time that you run a new FITC to that benchmark. And if those signatures are very different, if the similarity index is telling us, hey, those are different, then visually you can see we're going to give you a flag. If you have saved a PE and APC controls, and now those benchmarks are compared to what you're running on a daily basis, and they have a very, very high similarity index, good job, you're running the right control. So we have tools in the software to allow you to QC your control, and by you learning your signatures and understanding how to interpret your signatures, you will be able to QC your controls. We always check that the unmixing is accurate, as you will do in a conventional cytometer. Please check that your compensation is accurate, and if it's not, the question is why your controls are not giving you enough level of accuracy. We always check the unmixing first, looking at the single stain cells. If I use beads, are the beads giving me a good unmixing of the single stain cells? And then I go to the multicolor tube, because if you are trying to assess the unmixing accuracy directly into the multicolor tube and you're not familiar with the patterns, you might not understand what you are looking at. Whereas in this view, it's very black and white. You know if the unmixing or the compensation is correct. So do I need to run controls every single day? We have ways in the software that allows you to avoid doing that and to reuse the controls. If you plan to do this, please talk to us. Let us guide you through the process. Let's make sure that we're saving the best possible controls in the software so we get the best possible results when you reuse it. And going back to the single stain cells, the single stain cells are a key control, in my opinion. A lot of you say, do I really, really, really need to stain single stain cells? I want to stain beads only. Well, if you take the time once when you're optimizing your panel to stain single stain cells, you're going to get so much information and you're going to save so much time. You're going to have information about the antibody performance, if your titers are correct, if your staining conditions have been optimized, if your reference controls that are beads are going to be good for the cells. And on top of all this, they will tell you how good is your panel design. You need to take the time to compare each marker, how it behaves in a single stain tube versus a multicolor tube before you claim victory that you have a very good panel. Here is an example of our 36 color panel that we're currently, as I said, using in many different sites. And here is how the C45RA is behaving in the single stain tube versus a multicolor tube. And again, my eyes are with that, where this green arrow is, where the negative population is. What do I care? That the negatives are behaving similarly and that the intensity of the positives is very similar, meaning that the resolution has been preserved. There might be antigens for which and the top and the bottom are scaled identically. So this is key, right? I cannot compare resolution if the scales of my histograms are different. Where maybe there's more spread, maybe there is more spread in the negative population. But if there is more spread in the negative population, then can I live with that spread? And if I can live with that spread, that's fine. If I cannot live with this level of spread, if it's compromising my resolution, then I will need to go back and do some panel design. So here is the panel after we titer all the reagents, after we optimize the controls, after we check the single stains compared to the multicolor, after we went through all those layers, we started plotting the data. And it's beautiful, and we are so excited because we have this beautiful panel that gives us a lot of granularity on what's going on in the different cell types circulating in blood. How did we get there? Because we followed the principles and we took a step-by-step approach and we didn't take any shortcuts. Now, I'm going to ask you to always, look at this data in 2x2 plots. Absolutely not. We have very strong collaborations going on with different third-party softwares. FCS Express, OmiQ, Astrolabe. We're very grateful for all the work that they're doing to help us understand the data, to help us visualize the data. These beautiful UMAP representations are from OmiQ. What you have here is we run a clustering algorithm, flow sum, and then in the UMAP representation, we are seeing how these 40 different clusters are distributed, or we can do it in a density plot as well using the OmiQ software. And here we can track in how every marker in the panel is mapping. Again, in this with using the UMAP plots and seeing how, for example, for monocytes, we have 14, 11C, 11B, 141, etc. And it's beautiful, and you can get so much information. You can really have a deep dive into the data and a great understanding of the biology. Look at the resolution, look how crisp the information is. But before getting here, please QC your data and help us help you to get there. We can obviously do heat maps and look for those 40 clusters, which are the markers that are expressed in those different clusters, and get a lot of information about what each cluster is about. Now, it's great that we can do this, but we want you to be able to do this in your own lab. What is the way to get there? So we have a terrific team. I'm so blessed to work with everybody that is in the application support team. It's a great team of scientists, flow cytometry lovers, passionate about what they do. Please we have a lot of resources in the web, in our website, but being able to work side by side with you, allowing us to talk to you, to see some of the data is the best way to go, is the best way to get there, because we have been working on these panels, we have been characterizing the fluorochromes, we have a lot of knowledge and information that can save you a tremendous amount of time and headaches if you allow us to work hand by hand with you. Use the resources. Again, we have panels that have been optimized. We have panels that we have the OMIPS papers that are amazing and are a great place to start. But we all know that you want to modify panels. We all know that you have your specific interest. And so that's when, if we work with you, we will be able to get the most out of your assay. Here is an example of somebody who came to me and said, Maria, I need cytokines in that beautiful panel. Okay, no problem. Let's work together. Let's swap some markers. Let's make sure we use the best possible dyes for the cytokines. I love to use dyes that are small molecule that will give me little background. And let's make sure that we make the best possible decisions together in terms of panel design to incorporate that. If you come to me and say, Maria, 36 is not enough, I need more. Okay, I can work with you. We can incorporate more. We have beautiful dyes, 2 of them from Thermo Fisher, PLXA 610 and PLXA 700 that can be plugged on top of the panel. We have another BUV dye that was not used originally. So there are a lot of options, but we want to explore these options with you, and we want to make sure that we know from the beginning that we have a solid plan and a solid panel design. Thank you so much. I'm sorry it went a little bit long. Thank you for giving us this opportunity. Thanks for your attention, and we really look forward to working closely with everybody that is interested at having great data. And now I think there might be a few minutes that we can take for some questions.
Okay, thank you very much, Maria. There's been lots of great questions coming in, so I'm going to try and address some of the more common ones. So one is, for the panel that you showed today, how do we get help accessing and applying that panel? And do I need to retiter everything in it for my own application?
So great question. We have, of course, do our due diligence and titer all the reagents in human PBMCs. We believe, I strongly believe, that those titers translate to whole blood. Of course, there might be small variations lot to lot. So it doesn't hurt. I think we can provide you a very good place to start for a quick check of the titers. how do we work? You let us know if you want to try this panel. You get in touch with your local TAS or sales representative, and then we get the conversation going. We have a very, very close collaboration with BioLegend and Thermo Fisher. We try to help you getting your hands into these reagents, and we want to be with you or check the data once you run it to make sure it's giving the results that we are expecting. I hope this answers the question, and if there's a follow-up question on that, just type it in and we will get back to you after the webinar, for sure.
Okay, great. And another topic that came up a lot was on the Similarity Index. People are wondering, when is high too high? Is it when you get a combination where it equals 1? And are there different rules for if you're applying those dyes on populations that are mutually exclusive versus populations that are not?
Great question, and that question makes me believe that I went extremely fast into that beautiful slide of the complexity index. So our cutoff right now to say you can use 2 dyes in combination is 0.98. So that will be the upper limit when we're trying to say, are 2 spectrum signatures different enough? Now, remember that we are also using the Similarity Index to QC the controls. And in that, what we want to achieve is 1. We want to make sure that your control matches the fluorochrome that you have already stored in your computer. But I think your question is about 2 dyes. Can I use them in combination? The answer is the cutoff is 0.98, and the example that I had in the slide is FITSI and Brian Blue 515. And there was another part
for the mutually exclusive when you're
okay, so about panel design strategies. Again, if you are going to use them, mutually exclusive populations will be the way to go. If it's not possible, have you used them, these combinations that are very overlapping in markers that are co-expressed? Yes, I have. I have used APC and Alexa 647 in markers that are co-expressed in the same population. The key there is the antigen level of expression. And if you're going to use them in antigens that are very highly expressed, it's going to be messy. If you use them in antigens that are expressed at low levels, it's going to work. So that is why starting with the biology is such a key point, because if we know the level of expression, we know how far we can go in terms of panel design. I hope this helps.
Okay, I'm going to give you one more, and then the rest of the questions, don't worry, everyone, we'll get back to you after the webinar. We'll be in touch via email. So this next one is the panel that we showed today. There's lots of different markers, and they're spread across many different cell populations. But what types of studies or information is out there if I'm working on an application where I'm using many co-express markers and trying to build up a nice big high parameter panel like was shown today?
So we have designed panels like these, people who are more interested in deep dive into the T cells, for example. Some of this data is proprietary for the users who have developed. Is it doable? Totally. Are you going to end up with all the 36 markers co-expressed in the same cell? No. Some will be down-regulated or up-regulated depending on activation or differentiation. And we play with that as part of the panel design, as we play with mutually exclusive. So if everything is co-expressed, the level of expression is key. And where are those antigens going to be expressed in terms of the pattern of differentiation is going to be key. And then we sometimes in those cases when there are a lot of unknowns, we take a very conservative approach. And then I think maybe it will require a little bit more of optimization than if we know for sure how these antigens are going to behave, but it's totally possible. The likelihood of success of the first design will depend on how much you know about the biology. I think depending on the area that you're interested at, if you tell us exactly what kind of panel you're looking for, we can share with you some information and give you some ideas on how to approach those kind of panels. But yes, it is doable.
Okay, great. think that's all the time we have for questions for now. For everyone that we didn't get to, don't worry, we'll follow up with you after the webinar. And again, thanks for joining. Thank you, Gil and Monica and Maria, for all your help today. And we look forward to connecting with you in the future. Have a great rest of your week, everyone.